Cool. I think we are live. So I think welcome, first of all, to Julie and Seth. Thank you for joining me. We are doing the Catalyst DeSci Researcher AMA session, where basically researchers who are seeking funding on our platform Catalyst are able to join us for this call and talk a little bit about the research, what it means to them, and explain it in their own words. One of the sort of founding principles of decentralized science is this community involvement and sort of this breaking down of walls between researchers and the people that they're sort of aiming to serve with their research. And I think that these sessions are really sort of integral in that. And I think it's also really cool when you get to chat to someone who has a super niche knowledge about a very cool topic and you get to learn something from them. So I enjoy these sessions a lot. For those of you who don't know me, I'm Ella. I'm Molecule's lead scientific communicator. I'm coming to you live from our Berlin offices, and Julie and Seth are in New York. And they'll be telling us a little bit about Project Accelerate. So a little bit of admin before we jump into questions for them is, first of all, Catalyst is our decentralized research funding platform. It is democratizing research funding, allowing individuals to support projects that resonate with them. The decentralized model aims to rectify inefficiencies in current research funding apparatus. And then, of course, just another admin point is all opinions on this call are our own and do not constitute legal advice, and we do not verify the researchers' statements. They are their own. So just to get that over and done with. Okay, cool. I've introduced myself. One more thing, as I remember, is so this is a stream. Please post your questions on Twitter or on YouTube, wherever you're watching, and we can get the researchers to answer them. So yeah, think of this as an interactive sort of experience. I think these are best suited to be conversations. So if you have any things that you would like to ask, don't hesitate to pop them as a question. Okay. Today we have Project Acceleration, which is dealing with brain tissue replacement therapies, which I know when I first heard of it, I was like, wow, that's really cool. Seems very sci-fi. And then after being able to sort of chat to the research team, I realized that it's actually not as outlandish of a concept as I think I first maybe thought it was. And kind of, I think the moment it kind of clicked in my head was Tiago, who is unfortunately not able to make this call due to family commitment. But I was chatting to him. He's one of the lead researchers on the project, and he was sort of saying, if we're able to regenerate and regrow other tissues in the body, why are we not doing it for the brain? And then he said, well, actually, some people already are, but they're only looking at very certain areas of the brain. So it's kind of this area that is under-researched and presents a really interesting opportunity to sort of learn more. I would love it, Julie and Seth, if you could introduce yourself. Let's start with Julie.
Hi everyone, I'm Julie, so I started this research this year, earlier in New York, when I was kind of trying to explore what I can do to help longevity accelerate. I don't actually come from a bio background. I had a organizational science degree in George Washington University, and then I started a few startup projects. I created social apps. I actually worked for crypto for 2 years, and then before that, I was in robotics and semiconductor field. And so I always had a deep passion for deep tech related industry, and I also I had since a very young age, I started to get curious about longevity because I know that it's the right thing to do and it's the right path to like to lead humanity toward. So that's why when I went to Luzelu, I was so excited about like chatting with all the people working in longevity, and that's when I started to think seriously about stepping into the new field. So after that, I joined longevity biotech fellowship, and then I started to understand the bottlenecks of like different longevity problems that's stuck in at the current stage. And then I also try to identify what is the optimal or the best thing to do at this time stage. And then I settled down on replacement after reading the book by John Hubbard. So so that's also why or how I met Seth. I visited the lab in Albert Einstein Medical School, and then just like get to know um, what Seth has been working on, and then um, thinking that maybe I could also start a project in this field, gathering all the best talents. Yeah, that's a brief background.
Amazing. Thank you, Julie. I'm sure many of our followers know Zuzalu. Yeah, a longevity city. Seth, would you like to introduce yourself?
Sure. Yeah. Like Julie said, we met through my mentor John Hebert, who is really a pioneer in the field of brain replacement, and I got interested when I started my program in his lab. He taught some of my classes, and so I joined the lab because I was very interested in the concept of brain replacement and tissue engineering as a route of therapy. And so, yeah, I've been working in the lab for 2 years now. Been working mostly with projects that are related to cell culture systems, growing neuronal stem cells of different types, mostly focused again on cortical tissue, and then also analyzing some of the data through single cell RNA sequencing. And seeing what kinds of cells we're getting in culture and comparing them to the kinds of tissue that occurs naturally from fetal samples. So.
Super interesting. So you guys are both at the Albert Einstein College of Medicine, correct? I think I didn't mention that earlier.
Right. Yeah, that's right.
Awesome. So I also thought maybe you could give a little bit of an intro, Seth, possibly on Dr. Tiago Goncalves, because he is not here and he's leading the project. As I said, he's got a family emergency. But I thought maybe that would be helpful for our viewers to sort of understand the team behind the project.
Right. Yeah. So Tiago is another professor at Einstein. He works again pretty extensively in regenerative medicine and studies the ways that you can create functional connections using neural stem cells, and he also studies development in vivo using mouse models primarily through using very high tech microscopy techniques you can actually image a mouse brain while the mouse is alive by installing like a little window in the skull cap, and so you can really get a high depth look at what various therapies are actually doing and how they're integrating into brain tissue. So a lot of his research is centered around those kinds of concepts. This project being an offshoot of that.
Okay, super cool. Yeah, I mean, like you can make the most perfect brain tissue, but if it's not going to integrate into the rest of the brain, then
right.
not that useful. So I'm glad that we have Tiago on the team. Let's actually talk about what this specific project is about. Julie, I know that this is very close to your heart, so I'd love to hear you explain sort of what this project is about and what you're aiming to do with the funds raised on Catalyst.
Yeah, so this project is about trying to engineer one particular region of the brain, which is called ACC. And we are trying to, so like the whole vision is to engineer the whole brain from fetal tissue, which is a way to reverse engineer our brain. And it sounds like it's like based on the concept that we don't have to understand how our brain works, but we are able to just like being able to control and grow it from its very early stage. So it's like less expertise and less time required for us to really like grow the components, the parts of that of like what compose our brain, so that once our brain machine is more functioning, then we will able to have the replacement part ready. And ACC is of particular interest because it's highly relevant to a lot of the psychiatric disease. And we like after neocortex, which is already done in B therapeutic. Neocortex is like really responsible for lots of the intellectual processes in the brain, but we are trying to think of another like really important thing that makes us human, which is like we as human have emotion, which makes us apart from AI. So being able to engineer ACC and on one hand is able to cure lots of the psychiatric disease that are once uncurable before, but then first we're able to engineer that and then replace that for the patients through either surgical procedures or through providing drug testing platforms for psychiatric drug companies to test their products on. And the other thing is that we're able to like through doing this we're able to preserve what makes human, and we're able to preserve what makes our emotions so beautiful through like the molecular or cellular components of it. Yeah.
Amazing, thank you. I think sort of what makes us human bit is, I think, for me, when I first sort of looked at the research, is what really sort of struck me as unique about this project, because as far as I know, the ACC is a region that hasn't been regenerated, or other companies are not attempting to regenerate it. And the ACC is a pretty important part of our brain. And so I would love it if one of you could maybe tell us a little bit more about the ACC and, what it does for us, where it's located in our brain, that sort of thing.
So I guess I can provide a little bit, I guess, on that. The ACC it stands for anterior cingulate cortex. It's a region of the brain that's especially important for things like regulating emotion and sensory response and sort of contextualizing sensory responses in emotions such as pain, fear, things like that. And so it's like you said, it's not really an area of the brain that's been sort of a target of a lot of research with respect to therapeutics, and is pretty understudied. So it's definitely an area that's of interest from sort of a stem cell and regenerative medicine perspective, for that reason, because there's just not a lot of research in that area in general. So I guess, yeah, I don't know. Is there any?
Do you have any sense for why that might be? Sort of like why other areas of the brain have sort of been deemed more important?
Yeah, maybe. I think part of it is because some of the most common neurodegenerative diseases don't necessarily originate in that region of the brain in particular. I think more there are more it's more sort of a region of the brain that has more I don't know if subtle is the right word, but there's sort of more, there's different kinds of changes that aren't necessarily like that aren't necessarily like, you don't necessarily think of this region of the brain most readily when you think of diseases like Alzheimer's disease or prefrontal dementia and other kinds of diseases that are more geared towards other regions of the brain. So I think it kind of gets put on the back burner because of that reason, that there's these specific diseases that are more in other areas of the brain, but this, that isn't to say that this region of the brain doesn't have an essential function. It's just, it's not necessarily on the forefront of some of those other diseases.
Okay. And I think Julie kind of mentioned this, but like what happens when something goes wrong in the ACC?
So there's a number of, oh, sorry. Yeah, go
I
ahead.
will add on to it. So what will happen if anything goes wrong with ACC? Like it could cause diseases like OCD or depression and all those like psychiatric disease. Maybe Seth can add on to the processes of why ACC, if it's malfunctioning, the patient would have those symptoms or behaviors. But one experiment we know is that when a patient is deprived of ACC, when the ACC is not absent, then or not at the absence, then or like the patient lost their ACC tissue, then what happens is that the person is not able to do, like, it's not motivated to do anything, or like it's not even, like, the person just lost its ability to feel things and then just function as a normal person. So that means that ACC really is critical for like the fundamental motivation of humans when humans do a lot of things. So without the emotional motivation, they're not able to do intellectual stuff on top of it.
Okay, yeah, interesting. So basically, it's not so much responsible for our physical health or well-being, but our emotional well-being, which I think, is deeply important.
And then we also did some research on the market size. So why we choose ACC to work on is also because we know that psychiatric disease, this market has a 20 billion market size. So this is like a huge potential or huge commercial opportunities for us. So when we choose our region to work on, we want to choose something that makes sense for investors, makes sense for patients, and want to save more people from that.
100%. Okay, amazing. Thank you so much. I would love to know a little bit more about what this actual funding aims to achieve. So what's set up for the lab? What are you hoping to get out of? What are your endpoints that you want to reach with this funding?
So our lab is set up initially in Albert Einstein, and then after we fundraise, we will be able to maybe outsource some of our lab to grow its branch outside, or we will be able to set up a new lab if the funding allows. And then we are already doing some early stage ACC cell identification experiment where we're trying to just get the data of what makes the fatal ACC tissue, and then publish that data through a paper. So that's what we're already working on. But then after that, like using the money, we will be able to not only like know what grows the ACC and in different stage what are the cell components, but or extracellular components, but at the same time we will be able to like just engineer it in the right ratio, like cell density and like with the right structure. So it's like by the end of our research, we'll be able to generate very valuable IP, and then just like hopefully for the best, we'll be able to engineer ACC tissue, and then start to manufacture that, either customize it or to mass manufacture it for like our drug companies who are in partnership with us, or like start to explore clinical, like clinical or therapeutic treatment, like to go that direction. Yes, and maybe we can like have Seth to share about the findings of like neocortex, like the data we generated or like he generated from the other project. Yeah.
Okay. What I would love also just before we do that is I'm realizing that I think for a lot of people, like the actual like steps that go into regenerating tissue are probably not super familiar. So Seth, it would be amazing if you could kind of just like super simple forms explain how one would start with like a bunch of materials and end up with tissue.
Sure. Yeah, I can do my best to try and talk about it in common speak. But really, I think the process can be broken down into a couple of steps. The first being you first need to identify the particular components that go into the tissue you're trying to reverse engineer, and so the way you do that is you can study from you can take tissue samples from natural tissue, from natural fetal tissue or whatever tissue you're trying to reproduce, and you can characterize the different things that are there, such as the cellular components are characterized. The gold standard for characterization of different cell types is single cell RNA sequencing, which is a very high resolution way of assessing like what kinds of cells are actually present in your sample. And so once you can get a sense of like what cells are there, then you can do things like assess the proportionality of these different cells in the tissue, potentially assess their regional placement, and other kinds of things that are essential knowledge if you want to reconstruct the tissue. I will say that cells and cellular components aren't necessarily the end-all be-all for as far as tissue is concerned. You also need to understand sort of the extracellular components that are present as well, because those form instructive cues for the cells and the functionality of the tissue. So you can also use things like proteomics to figure out what kinds of proteins are in abundance in the different parts of the tissue. And so then you can use that knowledge to essentially try and mimic the right environment in a cell culture system, and try to grow those same cells from an early stem cell state into the cells that form that tissue. And so that's kind of the general idea. Of course, then once you have that kind of tissue prototype in place, then you can do things like surgical testing, to see what kinds of conditions are the most ideal for getting proper integration into brain tissue as a therapy.
Okay, very interesting. I'm just thinking my next question. I've got so many. I think maybe let's sort of talk about like how one would test if something like the ACC is truly, implanted and working well. And like, I mean, I think these sort of emotional metrics are much harder to track in like model organisms such as mice or something. So I'd be curious to know how that would happen.
Well, Yeah. So behaviorally, when you're testing anything with mice, it's difficult to assess sort of on a behavioral functional level whether or not your tissue is actually responsible for restoring a damaged circuit, simply due to the fact that that mice have a very high capacity to sort of to have the surrounding tissue. It actually will sort of sorry, I'm blanking on the right word here, but it's like the tissue will actually like restore the damaged function without if you don't, regardless of whether or not you add new tissue. So the compensatory function of the surrounding tissue is difficult to sort of separate from the effect of your transplanted tissue. And so given the timing of that, it's difficult to assess very, very complicated things simply using a mouse model. What you can assess is looking at sort of the gross anatomy of the integration of the new tissue. So you can look at whether or not they're forming synapses with the surrounding tissue, whether or not your tissue cells are surviving, and you know, if they're growing. Also look at their organizational architecture. So there are sort of more there are more indirect ways that you can assess things like that. And when you have evidence like that from mouse models, then you could potentially utilize it in more complicated models, such as like non-human primates or animals with a larger brain, or even maybe using it in certain clinical use cases where you know the patients, you know, don't necessarily have a lot of other options.
Okay. Yeah. Okay. Interesting. Yeah. I always find when chatting to researchers that one of the like more sort of unknown elements is like the amount of creativity it takes to design experiments to like actually get the data that you need. So I always sort of like to chat about that because, you know, at the end of the day, there are certain things that are difficult to test. And so I always enjoy hearing about creative solutions to it. So I think maybe taking a little step back is sort of what, I mean, I think, Julie, you're quite attached or very close to this project. I would love to know what it means to you, like personally, like why you're so driven to pursue this project.
Yeah, so what pushes me to keep doing this project is like first like it's just like very important. This is not yet possible technologically speaking. Like this is like we're trying to create moonshot, and then this kind of project has its own charm because it's challenging and it's like if you do it, if you actually do it, then it will create like massive impacts on so many things, and it will change the way people live their life. So like it's like fundamentally we're able to make longevity come true. We're able to extend lifespan by not just 3 years, 5 years, but 100 years, 200 years. So one thing that keep pushing me to keep doing this project is this sci-fi vision, and then it's like enabling so many like potential things, and we can imagine a whole new world with that. So yeah, and then like ACC, I think one personal reason is that my cousin had tumors. So when she was very young, she started to undergo brain surgery, and then I grew up from a very early stage to know about brain surgery, and I know that like how terrifying it sounds at first, but then eventually like every time it turns out her surgery is successful. So it's not like there's a very like high risk, or like it's actually it's actually very, very well established surgical procedures. So I think brain surgery as a concept may sound very far away for the majority of the people, but there are already like maybe millions of people, or not that amount, already like undergo that on their yearly basis. So yeah, I think both of those reasons make me like just getting more curious over time about it and wanting to like make an impact here.
Well, thank you, Julie. Okay, so before we move, I wanted to ask also, so Julie has a crypto background, which I always love to see in the DeSci space from coming from researchers. I wanted to ask a couple of questions about that, but before that, I sort of maybe just wanted to wrap up the project talk and sort of say, so first of all, the project is live on Catalyst. So if you are interested, go check out the project page. You can choose to support Julie and Seth's work and help them achieve their research goals. There are a bunch of materials there that are designed to help you dig a little bit deeper into the research. And yeah, I hope you enjoy. Okay. Julie, I wanted to know sort of how, like, what the experience has been as a researcher that has a crypto background, sort of dealing with Catalyst and DeSci, and sort of why you chose to take this route.
Um, so like initially, what led me to molecule is that I think it's before I started this research. I was still like wandering around in those pop-up cities and then trying to like commit to something, and then like one day I was talking to Vitalik, and then he created a group chat with me and Vincent, and then I was kind of pointed to Molecule, but I don't know if I should work for Molecule or what Molecule is. So I did more research after that, and then I realized that it's a like fundraising platforms, and because I was from crypto, so I like on one side I want to I know there are so many rich people in crypto who made their money before like 2014 or something, and then those money doesn't really like have a good place to go to. Like if you just keep funding NFTs projects and token meme coins, then it's not really a good use of the money. And I know that this crypto world also want to do something with a very good intention, and in the long run have a very good positive impact on larger population, or just like create those sci-fi technologies. So that's why I think what Molecule has been doing is really great, and it just makes sense. It just makes sense logically. It makes sense. Um, like I also know that there are many people who work in crypto. They're already starting longevity fund. They're already trying to like use their connections. They use their understanding of crypto to fund more longevity research and longevity companies, because it is the next thing. It is the next thing after crypto that's going to be that's currently more ignored, but then eventually it will be recognized by more people, and it will be the next thing as crypto has influenced many. Longevity will be the same. So yeah, I think Molecule is really great. That's why I choose to work with Molecule right now.
Amazing, thank you. Cool to know we're being passed around group chats. And yeah, thank you so much, guys, for trusting us with your project. We actually have a question, which is quite exciting, from Casey, who's asking, do you have to grow the new piece of tissue to the same size of what is being replaced?
Yeah, it's a good question. So So based on the knowledge that we have, that we don't necessarily need to grow the tissue to the size that of the tissue that's being replaced, because one of the so the idea here is we want to have a simplified fetal tissue structure that we can use to transplant into an adult brain site, or like a lesion site, and the idea is that the tissue will grow naturally to form those connections with the surrounding tissue as it grows and forms the mature layering structure. And the way that process works, usually it's not necessarily a matter of size or an initial size so much as sort of if you have the right conditions, the right growth conditions for getting the tissue to get to the appropriate size. And so one of the key details to ensure that happens is to make sure that the initial grafted tissue has proper vascularization, so that it can be supplied with the nutrients that it needs in order to grow and thrive and integrate. So I guess the short answer is no. You don't need the tissue to be the exact size of what's being replaced. But the longer answer is that we have yet to test sort of a variety of conditions and sort of maybe initial tissue sizes to see what is the most compatible. So that's. Yeah, I think that's the answer.
Okay, cool. Also, just a reminder, anyone else can sort of pop some questions in if they would like. I have a question, which is, please can you describe, so basically having read, the project description, there was also sort of this definition or sort of separation of organoids and proto-tissues. Casey says very cool, thank you. So thanks, Seth. Is this separation between proto-tissue and organoids. Please could you explain what that difference is and maybe give us a definition for each in simple terms, please.
Sure. Yeah. So organoids and organoid systems refer to really the growth condition that you use when you're differentiating the cells, starting from the earliest stem cell state. So what are called embryonic stem cells are sort of used as an initial starting point when you're used in most cell differentiation protocols. And so the way that you grow those cells to make an organoid is you grow them without attachment to a cell culture plate surface. So you grow them just sort of suspended in liquid media. Sometimes you grow them with orbital motion, and they form these aggregate clusters of cells that are sort of meant to resemble, in some ways, the 3D architecture of an organ structure. The caveat to using this system is that that isn't really the way that the tissue forms in the context of body development. So it's more sort of like it's more random in that you have these different cores of stem cells that sort of grow out and form the cells that you want, but they're kind of randomly interspersed throughout the mass or throughout the aggregate of the tissue. They're not super reproducible, or from different patient cell lines, or from different differentiation conditions, and so they're not necessarily ideal for a reproducible therapeutic. In contrast, a proto tissue and sort of the model that we're trying to pursue is more grounded or more centered around the natural architecture and biology. So our goal is to actually create a condition where we have the cells growing in the manner that they would grow in fetal development. So we want them to form, their actual uniform layered structures, sort of starting with like a flat layer of the the rudimentary stem cell components, and have them as they divide and differentiate, form layers on top of that initial layer, and in a uniform and polarized manner, and instead of sort of in a random heterogeneous manner. So that's kind of I think that's the main difference. But most research is, again, sort of focused on these organoid approaches because they are, in some ways, I think, more well established. I guess but their purpose isn't necessarily as clinically oriented because they're more focused on developing a rudimentary model for brain development and studying it in a more 3D context, but not necessarily as a transplant.
Okay, very interesting. So kind of like what I'm getting is that, like, on top of being like brain replacement therapy, which is, I think, just a cool topic, is like that the ACC is kind of under researched, and also the sort of proto tissue model is, or sort of pathway is also more overlooked than from what I'm getting. You say it should be. Cool. We've got a question from Su Yen, who was asking, how could you ensure the vascularization of the tissue?
Yeah. So one way, or the primary way that we've been variable that we've been adjusting for this is adding different proportions of endothelial cells or other kinds of vascular cells into the initial graft as it's put into the brain tissue. We know that if you adjust the proportions of these cells in an ideal ratio, you can actually get the cells to form to form vasculature in their own right, and they can actually connect to the surrounding vasculature in an adult brain, or into the transplant site. We've seen this in other regions of the cortex when we transplant just fetal tissue that we add additional vascular cells to, or cells that neural stem cells that we add into into a matrix scaffold along with vascular cells. They'll actually form a vascular. They'll form connections with the vasculature in the surrounding tissue that allows them to grow to a larger size than tissue without that initial those vascular cells in the graft. So that's one way you can seed the vascular cells into the graft as you transplant it. You can also add chemicals that can encourage vascularization, such as VEGF, which is used in some contexts to sort of pave the road, so to speak, in encouraging those kinds of vascularization processes.
Okay. Cool. Thank you. So we are kind of nearing the top of the hour. So I wanted to say a big thank you to both of you for coming and talking about your project. It's been super interesting. I hope that the people watching have also found it quite interesting, and have a new scientific fact to tell their friends. If you would like to support this project, check out their page on Catalyst. Julie's also active on Twitter, so you can give her a follow as well for more direct updates. But yeah, we're super excited to be supporting, these researchers looking for funding. And we will be hosting another AMA session for another project next week sometime. So if you just want to sort of subscribe to our Luma event, then you will get reminders directly to your inbox when we're having these chats with researchers. But yeah, it's been a great time. Thank you both. If you have any closing remarks or anything, you can do that.
Thank you for this, Ella. I appreciate it.
Yeah, thank you for attending our talk.
Amazing. Okay, cool. Everyone have a good evening, day, wherever you are. Stay safe and love to you all. Bye.